So this is sort of an overview of the chapter. It's focusing on how do you grow microbes? What do they need? And how do you manipulate them? And to understand this, when we cover this, it will integrate all of the things we've learned about their basic structure, their anatomy, the chemistry that is underlying all biology, and it will be a practical application of what we learned about metabolism last time. So just to define what growth is basically an increase in population size. So that means that there's a larger number of individuals. Now, if we were talking about humans, obviously that wouldn't be a larger number of humans. When we talk about cells, it's a larger number of cells. This is a pretty typical view of what you would see if you actually culture microbes in a lab. So this is a Petri dish, and we'll be using a lot of these. This one is 10 centimeters in diameter. That's the standard size that we will use, but if you've worked in other settings, you'll know that they can get really small and cute and really enormous. So, just so we all are on the same page with math and conversions, 10 centimeters is how many millimeters? 100, right? So it's just a tenth. This is a colony. We're going to define what that is in a second if you don't already know. But this colony, how big would you estimate it to be? If this is 100 millimeters across, then how big is that guy? 1 millimeter-ish. Yeah, so between 1 and 3 millimeters is the size of a typical colony. So in this particular picture, this is about a 3 millimeter colony. You can see smaller ones and somewhat bigger ones. As we go through the labs, you'll see a lot of colonies and you'll sort of start to get a sense of the difference in size between different species. The colony is a visible manifestation of the growth of microbes, but individual cells, are they visible or not? No. Individual cells inside the colony we cannot see, right? So we can use this technique to see them, but we're actually not seeing the individual cells because they're between 1 and 10 microns across. Okay, so what is the difference in size between a colony and an individual bacterial cell? Compare, let's say, a 1 millimeter colony to a 1 micron cell. What is the difference in size? Like, what's the sort of scale there? The colony is how much bigger than the cell? 1 millimeter versus 1 micron. What's the difference? Is it 10 times bigger? 100 times bigger? 1,000 times bigger? 1,000 is correct, yeah. So just to get you used to thinking about those units, again, because we're using smaller units than we're used to. Even if you've used metric units your whole life, then the micron and the nanometer and the picometer and the less than that, we won't go into that, but it is really helpful to feel very comfortable with those differences. So if that's the case, let's suppose that this is 1 millimeter and the bacteria that makes it up is 1 micron, then how many cells would you estimate there to be in a colony? Just a lot? Okay, so just give me a rough guesstimate. Say again? 2,000? That would be a good guess. It's actually more than that because 2,000 cells would fit in a tiny line you couldn't even see across the diameter of that colony, right? So it's going to be actually a lot more because not only do you have to fill all the space in the area, but the colony has three-dimensionality. So it's cells piled on top of each other. So it shouldn't surprise you that a typical colony has at least 1 million cells in it. Now, every single one of these colonies started with just 1 cell, right? So this represents a clone of 1 cell, dividing many, many, many times. And this is the basis of our being able to do pure culturing. Even though we can't see the microbes very easily to manipulate them, we can actually do very simple techniques in order to get pure cultures. So how long do you suppose it took to go from 1 cell that was originally here to this colony that contains 1 million cells roughly? Yeah, it depends on the division time of the individual species. But average, just an E. coli cell, for instance. Yeah, 1 or 2 days would be a good guess. So how did this whole technique of culturing cells really start? One of the pioneers was Robert Koch. a German physician in the mid-1800s. And you should have heard of him before. He was in the first chapter. What was his sort of claim to fame that we've already discussed? His postulates. What were those about? Yeah, he was linking diseases to specific causes and microbial causes. So that's very profound, actually, because, again, you can't see these as individual cells, although you can see them as colonies, which was a very helpful kind of intermediate way to get there. So he studied pathogenic bacteria. As we know now, there's lots of different kinds, and many of them are not harmful to us. But for his work, he won the Nobel Prize at the turn of the century. And he developed a lot of methods that we still use variations of today in order to grow bacteria. And one of the things he wanted to do was common to grow cell suspensions. You think back to those early experiments trying to determine whether or not spontaneous generation or biogenesis was going on. People were used to doing broths and growing things in broths. He wanted to do them on a solid surface. And he tried gelatin at first, which was actually a poor substrate because the microbes like to eat it. But one of his friends' wife was really into canning and jelly making, and she suggested using this thing called agar that she used to harden jelly, and that was very successful. We still use that today. So where are we now? We now have an understanding that there are these simple single-celled organisms in almost every terrestrial climate that you can think of, from the bottom of the ocean to like crazy hot acidic places like inside of a volcano, in the ice core. And it's thought that these extreme environments are actually likely to be more similar to the environments that we might find life on other planets if that is something that's ever discovered. Despite this diversity, right, because we know microbes can live in the climate that we live in, and also now we understand that they can live in all sorts of crazy places, each individual species is very limited in terms of what its growth conditions are. And they are also limited in terms of what nutrients they can consume and what they need to survive. Now, why do we want to grow them in culture? This should probably be fairly straightforward in some cases. It's very important to be able to culture them if we want to prove that someone has a particular disease or understand a pathogen better so we can treat it better. It's very important to have pure cultures if you're going to use it to make food or to make in manufacturing. And in research, of course, we can study microbes much better if we have clear ways to manipulate them. So what I'm going to talk about today is basically I'm going to address 4 questions. The first question we're going to talk about are what are the principles of microbial growth. So there's some key concepts that we need to understand. And initially you'll just have to memorize them, but there are mathematical logic-based applications that will help you kind of get a better sense of them actually more intuitively. Now we need to also talk about what conditions we use to grow microbes, how we culture them in the lab based on that understanding, and how we measure whether or not microbes are growing and how fast. This is extremely relevant to the next chapter where we're going to talk about how we use all of this information to control the growth of microbes in order to prevent food spoilage, in order to deal with infection, to study the environment, and to use them in industrial settings. So let's first focus on this. What are the principles of microbial growth? So first of all, how do we increase the population size? Single-celled microbes divide asexually, although there is a certain amount of sexual reproduction, the so-called sexual reproduction that happens in the prokaryotic world. That's the horizontal gene transfer, and you may recall we talked about piluses, right? There's a specialized kind of pilus that can transfer DNA between neighbors. And that's a way to sort of mix the genomes of 2 different organisms that are next to each other in much the same way that we mix our own genomes every time we have a new generation of humans. But by and large, prokaryotes reproduce clonally. That is, they have their own genome, and they just make an exact copy of it and pass it down to the next generation. And a lot of them do this, binary fission, although this is not the only way that reproduction occurs in the prokaryotic world. So just think about the difference between this and the sexual reproduction that we see, and which happens both in multicelled organisms and in some single-celled organisms. In particular, budding yeast, which makes what? What is the yeast doing for us? Yeah, it makes a lot of our food that's delicious. That budding yeast is very well studied for its sexual reproduction, even though it's single-celled. It has two mating types. Most people find that surprising. And they can actually reproduce and mix up their genomes. So here are some of the other ways that reproduction can occur besides binary fission, which is by far the most common. It happens in most bacteria and in some yeasts. There are yeasts that do budding. So the yeast that makes beer, wine, and bread does this. And basically, it's a very similar process to binary fission in that you just make a copy of your DNA, but then you make a little bud that grows out from the side of the cell instead of dividing straight in half. So it's kind of a cool process. I studied it for 6 years if you ever have questions about that. Some of them have spore-based reproduction, and some of them actually just break into filaments. We're going to focus on this method primarily. So it's pretty simple. I think this diagram explains it, plus you already probably knew everything you needed to know about this process. Basically, you just make an extra copy of your DNA. You actually attach it. You kind of attach it as if there's a little hook here to each side of the cell, and you divide it in half and build a new cell wall in the middle. Then the cells separate. Binary fission results in what we call exponential growth, in that every time this cell divides, it causes the population to double, from 2 to 4, 4 to 8, 8 to 16, and so on. And that's the definition of exponential growth, when a population doubles every division, and if that division is regular, we can measure it and make predictions about how big the population is going to be at various times. This is a term, the generation time, is the time that it takes for that doubling to happen. So in prokaryotes, for instance, the generation time is relatively short. If we were going to apply this concept to humans, how long is the generation time in humans as a minimum? In people, what's our minimum generation time? Like, basically think about how old you have to be to reach sexual maturity. What's the first possible earliest age you could have kids? Yeah, I mean, depends on when you reach puberty, but it's measured in decades, okay? And in prokaryotic world, generally speaking, we're measuring the generation time in minutes. This does vary among species, as we've already discussed, and it is affected by the environment. So depending on how optimal the environment is for the cell, it might slow or speed up the generation time. But E. coli, for instance, at optimal conditions, has a 20-minute generation time. Now that is pretty staggering. Just think about how quickly that population can grow as long as everything is ideal. This is the basis of the 4-hour rule if you've ever worked in food service. Which is what? Does anyone know what this 4-hour rule means? Yeah, it's how long food can stay out, not be refrigerated, and still be considered safe to eat. That is perishable things. Obviously, it doesn't apply to, say, your tomatoes and bananas, which can sit out for longer. But cooked food, that's supposed to be cooked. Meats or mayonnaise or something like that, that's perishable. So that means that food can sit at room temperature no more than 4 hours. We can do things to it, though. We could put it in the fridge, and why is that helpful? It stops or slows growth, depending on the growth requirements of the species that we're controlling. But here's an example. So 10 cells of some pathogen in food. Within 4 hours, if the conditions are ideal, which say room temperature and whatever food source it's in are ideal conditions, will result in 40,000 cells in 4 hours. So we can actually calculate this number. It's not just sort of something you have to know. And we use this equation. If you don't like math, don't be scared. We're going to walk through this. So this is the number of cells total, which is calculated by taking the initial number of cells and multiplying by 2, which is doubling, raised to the power of x, which is the number of divisions. So how can we figure this out? Let's take an example of a potato salad at a picnic. And let's suppose the potato salad initially has 10 cells in it. These 10 cells have a 20-minute generation time, much like E. coli. So what is our N0? 10, okay? What is our X, the number of divisions? How do we figure that out? Well, we have to know the generation time, 20 minutes. And we have to know how long we're going to let it sit, which is 4 hours. So if the generation time is 20 minutes and we're going to let it sit for 4 hours, how many divisions is that? It's 12, right? So you just plug all of this in, and your final number of cells is 10 cells to start with, doubled 12 times, which gives you 40,000, actually roughly 41,000 cells. Now, an analogy for exponential growth that will illustrate, I think, powerfully how quickly things can change when they're exponentially changing, and how sort of counterintuitive it is that example of you win the lottery and you have these two options and you have to decide which one you're going to choose. So if the first option, you will receive $10,000 every day for a month, say the month is 30 days long. And the other option, you will receive a magic penny that doubles in value every day for 1 month. Now, just off the top of your head, which do you think, and be honest, okay? You probably know where I'm going with this if you have, if you know what plot is like, but be honest, if you had these options, what would you think would be the best?
Option 1.
Option 1. One reason option 1 seems good is because $10,000 even on day 1 is pretty awesome, and it's very easy to calculate how much that is, right? This is a sort of an increase by addition. So every day we add another $10,000, and at the end we have $300,000. Everybody gets that, right? All right, so let's look at the penny that's doubling every day. Let's look at the first 10 days. Okay, so day 0, we have 1 penny. By day 5, we have 32 pennies. Okay, that's not very exciting. On the 10th day, we have 1,000 pennies, which is worth how much? $10. So I'm sure at this point you'd be like, I'm a little nervous. I wish I had taken the $300,000 like my friend. Day 20, you've got 1,000,000 pennies, which is still only $10,000. So 20 days is how long it took you to get to where that person got on day 2, right? I'm sorry, on day 1. Now, what do you think happens at the end of the month, though? When we take 2 raised to the power of 30, what is the total? Basically, one way to figure this out without having to do crazy math and not using a calculator is that every 10 doublings, you're increasing the amount by 1,000. Okay, so here's the first 10 doublings, you're increasing your amount by 1,000. The next 10 doublings, that's 1,000 times 1,000, which is roughly 1,000,000. And if we take 1,000 of that to get to day 30, then we're getting to 1,000 million. Now, how much is 1,000 million in pennies? In dollars? It's $10 million. So I think at that point you'd be pretty happy if you made that choice, or sad if you hadn't. All right. So the problem with exponential growth versus this kind of addition type of growth is that the numbers really rapidly get out of hand. So if you're looking at, say, over a relatively short span of time, like just 20 units, whatever days, hours, whatever it is, you can go from 1 to 1,000,000 very rapidly. So graphing that is a big pain because where you put your axis, like where you choose your scale, you're only going to be able to see resolution of those numbers for a short period of time before it either gets too small or too big. So instead we can take the log. And the log basically tells us something kind of about this number. Actually, the log is, how would you get this number if you just took 10 raised to the power of something, right? So 100, the log of 100 is what? How many times do you have to multiply 10 by itself to get to 100? Twice, right? 10 times 10 is 100. Does that make sense? Okay. So if you take the log of 100, you get 2. If you take the log of 1,000, you get 3. So your numbers go much more slowly. Think back to pH. pH is measured on a log scale as well, right? So every time you change your pH unit by 1, you actually change the number of things by 10. And you'll remember that annoying question on the exam about pH. pH 2 versus pH 4. What's the difference in terms of the concentration of hydrogen ions? 2 to 4 is 100 times different, not 2 times different. Yeah? Okay. So here's the difference if you're graphing. So on the left, these numbers are shown on a log scale, and on the right, they're shown with real numbers. So if you're trying to graph this population using real numbers, then for the first 15 generations, there's barely any change on this particular scale, because we had to make the scale really huge in order to accommodate this final number. And consequently, as you go from, say, 5 to 10 generations, it's really hard looking at the graph to appreciate what the difference really is. But we can do it on a log scale, and we get a straight line that gives us extremely good resolution for these numbers. So, the last thing I want to say about this, if you're worried about how much you need to understand, especially if math is not your forte, is you do need to understand how to do this math here. But again, don't get hung up on trying to memorize this. Use your common sense to make sense of it. And the questions that I would ask on a test would not be ones where you'd have to plug in huge numbers on a calculator. Right? Just imagine that what you're doing is you're doubling your population in a regular period of time. So you may have to do things like figure out how many times you double your population based on the generation time and the amount of time that you're going to do this in total. And then just take the starting number and do a fairly simple calculation. There are some examples for you to use in the supplementary module online. If you do those questions and you're still confused, then we should talk so you can do well on the test. It's not going to be, a huge number of questions, just 1 or 2, but if they're there, you want to get them right. All right, so now let's talk about the phases of growth, because when we grow bacterial cultures under ideal conditions in a closed system, then there are certain very predictable things that we see. Now, what is the difference between growing bacteria in a closed versus an open system? What's a closed system? What would the difference be? A closed system is one where you don't get any more what coming in. You just have your environment, right? Like being on planet Earth. And we only have so many resources, and if we run out of them, or if we contaminate the environment with toxins, what's going to happen? We'll starve to death, or die. So, in a closed system, what we see is, if we first take some cells from whatever source, and we put them into an ideal environment, for the first little while, we don't see a change in the number of bacterial cells, which is graphed on the y-axis. So we see basically no change. We call that the lag phase, right? It's just waiting. We're going to talk about why that's happening. Even though this is ideal and there's plenty of food for everybody, there's no change in cell number. At a certain point that changes, and then we get exponential growth. Again, because we're graphing this on a log scale, we get a straight line. If we were graphing this using regular numbers, we would see this hyperbolic curve. So we see this very regular, steady increase in the number of cells, regular doubling, so you could use this to figure out how, what the doubling time is. And then, at a certain point, we reach stationary phase, and again, although we now have lots of cells, more than we started with, we now get no net growth of the population, and we finally enter the death phase, where the number of cells goes down. And I just want to point this out, and I will try to point this out several times. What do you notice about this decrease? It is also exponential, right? But usually not as steep as this increase. That means that if you are in this phase or if you are in that phase, you can predict with very good accuracy how many cells you will have at any given time point, and I would ask you that on a test. So yes, this entire thing is graphed on a log.
Yes, but it was exponential.
Yes.
Okay.
Yeah. So if we were just doing real numbers,
Yeah.
then this would look like a hyperbolic curve
Yeah,
instead of straight line, and this would also look like an inverse hyperbolic curve.
but they just do that.
So it's much easier to look at the numbers, yes. So one thing I want to mention, and I probably am going to remember to say this again, but there is actually a pretty fundamental difference between this phase where no growth is happening and this one where there's no apparent change in this population size. Here, the cell number is not increasing because, by and large, the cells are just not doubling. They're not dividing. In stationary phase, that's not true. In stationary phase, what's happening is that the number of cell divisions is equal to the number of cell deaths. And we're going to talk about why that is. So let's look at each of these phases. So lag phase was the first flat part of that line, where we put some cells into a fresh environment with lots of food, perfect temperature. They should be very happy, and yet they don't actually double for a while. This number of cells does not increase. And that is because they have to adapt to their new environment, right? They have to sort of look around, take stock of what carbon sources are available, and maybe they've come out of a saturated culture where they were kind of hunkered down, just trying to survive all the toxins. So they have to sort of recognize where they are, and they have to make proteins that will allow them to take advantage of that new environment. So the lag phase time can vary depending on species and depending on how different the new environment is, but there's always this wait. From the lag phase, we go into log, and don't get these mixed up. I will test you on this, and I'm sorry that it's only different by one vowel. There's nothing I can do about it. It's also called the exponential phase, but most people refer to it as the log phase, so I want you to be comfortable with that. And in log phase, cells are dividing at a constant rate and allow us to measure generation time. So again, this is because there's plenty of food, the environment's fairly ideal, and the cells are just dividing at a regular pace, right? So they're going to divide every X minutes for that whole entire period of time. Now, this phase is one where the cells are the most sensitive to antibiotics. Do you have any idea why that might be the case? This is counterintuitive for a lot of people because they think, oh, there's lots of great stuff around. These guys should be super healthy. And we sort of think, like, when we're super healthy, we can ward off infection better. Yeah, they're making new stuff all the time. And we haven't talked about every possible antibiotic, but we do know about penicillin. And what does penicillin target? It targets peptidoglycan linkages in the cell wall, which are going to be made new, and it specifically targets an enzyme that's responsible for making those. So it's going to be inhibiting the synthesis of new stuff. So from here we go to stationary phase, and again, what's happening in stationary phase is basically that by and large you've run out of nutrients. Also, you have probably started to put some toxins into the environment. And what's happening now is that some of the cells will die, and when they die, they release their contents that provides nutrients for neighboring cells who then can divide. So it's in a perfect balance between this cell divides, but it produces enough food to allow this cell dies, allowing this cell to divide, but the death and the growth are equally balanced. And then we get to the death phase. So sad, this has to happen eventually. The total number of viable cells starts to fall. They die at a constant rate. And I just want to emphasize this because I think a lot of people don't recognize this, and it is important to understand. In this phase, again, we see an exponential decrease, and usually a slower rate of decrease than we saw during the growth, but nonetheless, it is exponential. Anything that kills cells does so exponentially. And what's interesting also about this phase is that some fraction of the cells may survive, so not everybody's killed. And if they survive, it's frequently because they have adapted better to the worsened conditions around them, which actually will often make them more antibiotic resistant, more resistant to the toxins in the environment, more resistant to a variety of things that otherwise would kill them. So while their friends and neighbors are dying, they're actually getting, in a way, they're getting stronger. All right, so let's talk about colony growth. So all of that was in liquid culture. That's sort of assuming that you have, you have some kind of closed system, and you're shaking it to provide enough oxygen, and you're kind of making sure everything's evenly distributed. But in a colony, that's not really the case. If we think about what is happening in a colony, let's see if we can diagram this. So here's the surface of a plate, and here's a single cell. And he's going to start dividing and dividing and dividing. And pretty soon, there are some cells that are at the outside edges, and there are some cells buried way, way deep inside. Okay, so which one of those guys is going to have access to the most oxygen and fresh nutrients? The guy on the outside edge, right? He might be a little lonely, but he's going to be in a better position than the guy buried way deep inside, who's got more and more cells piled on top of him, definitely is becoming anaerobic if oxygen is important, which is not important for all microbes, but if that's the case. And certainly, if you're surrounded like by all these cells, so this is now sort of side view, here you are, then any toxic byproducts, everyone is basically secreting toxins onto you. This guy has, a whole side where he is not affected by that. So the microenvironments in the colony are quite different depending on where you are. So again, the edge of the colony has the greatest oxygen or gas exchange, whatever that gas is, and nutrients, whereas the center of the colony is depleted for gases and nutrients and is also a place where potentially toxic waste products are accumulating. So as a result, within the colony, you can have different parts of this growth curve happening simultaneously at different locations. All right, so now let's talk about what conditions microbes need. What things do we need to consider, do you think, when we talk about this? So we have to worry about nutrients. What else? Temperature. Anything else? pH. Other things? What about gases? All depending on what kind of conditions these microbes like, that can be important also. So we're going to talk about a variety of things, temperature being one of the most important, pH. Now water or osmotic pressure is important, and we're going to sort of go into a little more detail about what specific terms mean when we talk about this requirement for water. Also, we need to consider their oxygen requirements or their need for detoxification enzymes, and we're going to discuss why you would need those. Nutrients, of course. So first, looking at temperature, we have a chart here showing different categories of organisms based on which temperatures they prefer. So what you can see is that there are some organisms that prefer cold temperatures on the left, shown in blue, and some that prefer really hot temperatures and others in the middle. But each one is a narrow range, right? So it's not like there's one organism that has a broad range and can survive at -10 and 67. We can group, and people frequently do classify organisms on the basis of their temperature preferences. This is super obvious, but I'm just going to say it. You can classify things by their minimum growth temperature, which would mean what? The lowest temperature at which they can survive. Optimum growth temperature? The best temperature, right? Which temperature is going to be best for them. And the maximum, which is the temperature at which they will still be alive. After that, they'll be dead. And usually the optimum is close to, but not at the top, right? Why is that, and why are the graphs shaped in this lopsided way? Why is the preference kind of a slow increase on one side and a very rapid decrease on the other side? Right. So as we go up in temperature, we're letting the enzymes work better and better. But at a certain point, that all falls apart because we heat the protein up too much. Now, why is it, do you think, that different organisms can have these different temperature requirements? What are they doing to survive at low or high temperature? They're adapted to those temperatures. What specifically are they adapting? But how is that happening? Think about it at a molecular level. What are they actually doing to allow, the cold adapted guys, how are they adapting? What are they doing to adapt? The high temperature guys, how are they adapting? Remember that the key to the temperature requirement is protein folding, right? So you could imagine that one way to adapt to colder or hotter temperature is to change the amino acid sequence so that you still have the same structure, but either that fold is looser or tighter, right? You would want it to be looser if you lived in a cold environment and tighter if you lived in a hot environment, because you either have to allow it to have more motion even though it's really cold, or allow it to hold on tighter even though it's really hot. Does that make sense? Okay. So here are just the definition of these different categories. Psychrophiles, that is cold loving, range usually between -5 to 15 degrees and are found in polar regions. We have cold liking psychrotrophs that usually optimally are living best between 20 and 30 degrees. And the majority of food spoilage organisms are found in this group. If we keep going, we have the mesophiles, right? Their optimal range is usually between 25 and 45. And of course, it should come as no surprise that human pathogens fall into this group, and those are a subset that grow best between 35 and 40 degrees. Why? Because human body temperature is 37. And then we also have thermophiles that are heat-loving. 45 to 70 degrees is sort of the range you can find them in. They're found in hot springs. And we also have hypothermophiles found, for instance, in ocean hydrothermal vents. And their temperature preference is quite high, usually, though not always. They're part of the group Archaea. So just to sort of show you an application for this, if the psychrotrophs are the ones that cause food spoilage, why is it that that's that group and it's distinct from, say, mesophiles that grow in the human body? Because where are we storing food? We're storing food either at room temperature or even sometimes in the refrigerator, and so organisms that can grow at those temperatures are going to actually be the ones that are dangerous and to our food. Are the food spoilage organisms usually the same as pathogens that make us sick? No. But if we're spoiling food, then we're probably also potentially growing pathogens at the same time. If we're in conditions where the food spoilage organisms can grow, there's quite a big overlap in terms of, the pathogens, while it's not maybe their ideal temperature, are still going to grow. That's why E. coli can still grow at room temperature and cause us a problem. So most of this we've already discussed. One point that I want to make is that these colder loving organisms grow more slowly because that's just a property of life. Their enzymatic reactions are going to go more slowly. And we frequently preserve food by freezing, but freezing doesn't kill the microbes, right? Usually it stops the ones that we're interested in stopping. That is the food spoilage organisms. They cannot grow, but they may not die, actually. They may just sort of go into stasis. The other thing that I think is really interesting to sort of pay attention to is this idea of the specificity of temperature in different pathogens. So a lot of microbes have this really narrow range, and their optimal growth is, a few degrees actually makes a difference. So if we think about a couple of different pathogens that are in the same genus, right? Genus is Mycobacterium, and in that genus, what do we have? It's an acid-fast genus. And it includes what? Common, extremely common, tuberculosis. So Mycobacterium tuberculosis is in that genus. A close relative, Mycobacterium leprae, causing leprosy. These two have slightly different temperature preferences. It's only differs by a couple of degrees, but Mycobacterium leprae grows optimally at slightly cooler temperatures. And that is why it affects the extremities, where our temperature is slightly lower. Okay, so now let's move on to pH. Most bacteria grow between what pHs? 6.5 to 7.5 roughly, right? Really close to neutral. There are some molds and yeasts that prefer an acidic pH, slightly acidic pH. Why is pH important? Because it affects protein folding. And remember that when we look at a pH curve, here in the middle would be the optimal pH for whatever organism. And then we see these sharp drop-offs on either side that are pretty symmetrical, unlike the temperature. Yes. Yeah, we'll talk about those in a second. There are some organisms, not very many, that prefer either basic or acid conditions, actually. So pH has this different look to the growth curve, and so you would be able to tell that apart on a test as a hint. So just to remind you what pH actually is, it's the negative log of the concentration of hydrogen ions, and it's a log scale. So when we say something's pH 2 and something else is pH 4, what kind of difference in the concentration of hydrogen ions are we talking about? When we go from a pH of 2 to pH of 4, is it a difference of twice as many or half as many? It's 100-fold difference, right? Because every time you go from one pH unit to the next, you're changing the concentration by 10-fold. Now, if we go from pH 2 to pH 4, are we going to a more acid or a more basic situation? More basic, right? Which means what's happening to the concentration of hydrogen ions? pH is going up, so hydrogen ions are going down. By how much? 100-fold as we go from, say, pH 2 to pH 4. If we went from pH 2 to pH 3, how much are we changing the hydrogen ion concentration? By 10. Okay, so bacteria have an optimum range of pH. Usually it's in that range somewhere in between 6.5 and 7.5, but there'll be an optimum that's unique to that species. And what they mostly do is maintain a constant internal pH that's neutral by just adjusting where they pump protons. So they'll pump protons out if the inside is getting a little bit acidic, and they'll bring protons in if their environment's getting a little bit alkaline. Most of them are neurophiles, so when we say neutrophile, we're talking about pH preference. This is sort of the broad range of this class of organisms. We can usually preserve foods by increasing acidity. So what kinds of foods are shelf stable because they're more acid? Say again? Yeah, so those canned foods have also been pasteurized in a special way that's more so than regular pasteurization. They've been tyndallized, basically. But they do have acid in them, and that helps to preserve them, right? Because an acid pH discourages most organisms from growing. Pickles, right? Relish. Most condiments are a little bit acid. Helicobacter pylori is this organism I mentioned in the first class, which causes what? Stomach ulcers. Now, obviously, the stomach has what kind of pH? It's acidic, so it must be adapted to an acid environment. How does it deal with that? Just as an example, it makes basically it does a couple of chemical reactions, and it produces urease that it then splits into carbon dioxide and ammonia. And ammonia, you may know, is very basic. So it's basically, it's in an acidic environment, but it's making a base and secreting that base outside so that it's on balance. It itself, as an individual cell, is in a neutral environment. There are acidophiles that prefer a pH below 5. There are, for instance, this one that was isolated, Picrophilus ashame, isolated from a Japanese volcano, that prefers a pH less than 1. And if you put it at a pH of 4, it'll die. It can live in 1 molar sulfuric acid, which I just find astonishing. And to answer your question, Ariel, there are alkalophiles with an optimal pH of greater than 8.5. So now let's move on and talk about water and osmotic pressure. So obviously water is really important. What kind of chemical reactions require water? And what's sort of the broad category of those? What do we call that? Hydrolysis, right? So to break stuff down, we need water. We also need water to dissolve things in so that we have a cytoplasm that things can move around in. But it matters what's dissolved in the fluid that you're in. So if there's a lot of salt and sugar, for instance, in your environment, that might be helpful to a certain extent to provide nutrients and trace elements, but at a certain point it becomes unhelpful. Why? As we increase the amount of solutes in the environment, what's going to happen to the water in the cell? What do you think? It's going to leave, right? Dehydrating the interior, then your chemical reaction shut down and you don't have any more cytoplasm. Now, how can we define this? Most cells, if you just look at sort of the broad spectrum, what's dissolved in them is about 1%. The solution is considered to be about a 1% solution. So for comparison, a 1% pure solution, say, of sugar would be 1 gram in 100 mils. So this is taking like basically 1 gram of all the things in about 100 mils would be 1% solution. If you are a cell and your contents are about 1% dissolved, and you put into a solution that's 1%, whatever it is, then you're in an isotonic solution, right? The amount of solutes on the outside are balanced by the number on the inside, even if they're not exactly the same chemical. The water is going to move kind of evenly and isn't going to want to be in one place or the other. A hypotonic solution is one in which we have high osmotic pressure because water is wanting to come into the cell and explode it. So in what circumstances is that going to happen? If we are a 1% sort of internal in our cytoplasm where our solution is about 1%, what's a hypotonic solution? That's referring to the outside. Is it less or more than 1%? Take a cell and drop it in pure water. Is that hypotonic or isotonic or hypertonic? What is it? What will the water want to do? It's going to want to come in. Okay, so it's a hypotonic solution. So if the outside world has a lower percent of solutes, that's hypotonic because the water outside is going to want to come in. Hypertonic means that the outside world has a higher percent solution, which means what is going to happen to the water in the cell? It's going to want to leave. Hypertonic means that the water leaves and you are dehydrated, and we call this plasmolysis, meaning that the cytoplasm gets decreased because water's leaving. And this is why we can preserve food by adding salt or sugar to the environment of the food, because the water inside the cell is going to want to rush out. It's also why things like Twinkies never die. If you've ever wondered, that's the reason. Now there are some obligate halophiles. What do you think this means? Obligate means what? Have to, must. Halophile, loving what? Loving salt. So marine bacteria have to live in about a 3% solution. This is different, right? What is normal? Like your average cell, what's normal? 1%. So marine bacteria need to be in about, their isotonic situation would be about 3%. Because they're adapted to that higher salt environment. And there are extreme halophiles that survive and actually prefer greater than 9% of salt. So those would be the ones that are adapted to, say, the Dead Sea, the Mediterranean Sea, or the salt flats in Utah. There is a group called halotolerant that is sort of in between your normal sort of regular cell type and your obligate halophiles. And they can withstand, they don't prefer it, but they can handle up to 10%. Okay? And that includes a lot of this species in the genus Staphylococcus. What species in this genus do we know of that you've handled in this lab? Or that you've heard of? Staphylococcus what? Aureus. And you've in this lab handled something called Staphylococcus epidermidis. Where do you think those organisms live? They live on the skin. Why would it be important for them to be salt tolerant? Because we are salty. Exactly right. All right, so let's move on to oxygen. The need for oxygen is complicated. Okay, so when we think about oxygen, what do we usually imagine? Do we think it's an important thing, not an important thing? It's important. Why do we have that bias? Because we need it. Do all prokaryotic cells have the same requirement for oxygen that we do? No. Now, many of them do, and the ones that we often care about the most, being pathogens, frequently do. Are there human pathogens, however, that are not aerobes? Yes. For instance, Clostridium tetani and botulinum, right? They form these spores. They're anaerobic organisms. They can't live in oxygen, but they can form spores that can live in oxygen. And then if you get that spore deep inside a cut, it could give you botulism. If you get it in canned food and it germinates and makes toxins, you can get sick from that. Now, we need oxygen why? What do we use it for? We spent like 2 days talking about this. Aerobic respiration, which is the primary means by which we metabolize sugar and keeps us alive. Microbes, some of them do use this, some of them don't. For the ones that don't, how do they survive? How do they get by? What metabolism do they do? If they're not doing aerobic respiration, what are they doing? Anaerobic respiration is a possibility. Some of them do that. What else? Yeah, they might be using like nitrogen or a sulfur or based or carbonate or something like that. If they can't do a respiration at all, what would they be doing? Fermentation. Now, it's thought that the early Earth had very little oxygen and that most microbes alive then would be killed in our modern environment. Because genetics shows that the strict anaerobes that are still living on Earth are more ancestral. And we have some very famous anaerobes kicking around, like the Clostridium genus includes a lot of anaerobes that make spores that can be pathogens. How do we determine these oxygen requirements? This is sort of a simple way. There are more complex and sophisticated ways. This is the one that you will do in this lab, not today. Basically what you do is you make, this is a solid agar, and you make it in a special way so that you drive the oxygen out of the bottom of it. Okay, so you drive the oxygen out, and you create a gradient in this tube, where at the bottom of the tube there's no oxygen. It's an anaerobic environment. And at the top, obviously, it's aerobic because it's in contact with air. And there's a gradient as you go through, like oxygen can diffuse a short way into here. So in the middle, how much oxygen is there? A medium amount, right? No oxygen, a little more, a little more, a little more, lots. So if you stab right to the bottom with some organism and let it grow, where it grows is going to tell you what kind of oxygen requiring organism it is. So this would be an obligate anaerobe, meaning what? Obligate aerobe means it needs air. An obligate anaerobe means it will be killed by air. Facultative anaerobes can grow in either environment, but they actually prefer an aerobic one. Why would that be? If they're capable of doing metabolism without oxygen and with oxygen, what's happening?
They probably prefer oxygen receptor, but they can use other things.
Yes. And when they use oxygen as their terminal electron acceptor, what happens? They grow quicker because why? They get more ATP out of every glucose. So, a facultative anaerobe, you'll see more growth at the top, but you'll see growth all the way to the bottom. Now, there's these two other guys, and we'll talk about this one first. There's microaerophiles that prefer less oxygen, but not zero oxygen, and a little bit of carbon dioxide. So they're going to grow in the middle and not at either end. And then we have these aerotolerant anaerobes. What does that mean? Okay, what's the difference between these guys? What do you think is happening here? What are they doing in order to survive? They're aerotolerant, meaning they put up with oxygen, but they're not actually making use of it. So what are they not doing? They're not doing aerobic respiration. Okay, so instead they're doing something else, fermentation or maybe anaerobic respiration. And because they're not doing this, they're not making use of oxygen. They don't have this profile where they grow better in oxygen. Because they're going to make the same amount of energy no matter what environment they're in. Now, why are some organisms killed by oxygen? Because oxygen's toxic. So there's a couple of things that you can make. If your oxygen is at the bottom of the electron transport chain and you're throwing electrons onto it, you can make toxic, or we call them reactive oxygen species. So if you stick an electron on oxygen, it actually makes a free radical. Have you heard of free radicals? Anyone? Antioxidants, have you heard of those?
Yeah.
So this big deal about you create these are toxic because they're very reactive. This guy, if you don't do something about him, he's going to just crash into everything in the cell and destroy it and damage the chemicals that make up the cell. And so you would imagine that if you have lots of free radicals, you're going to, say, age faster because your cells are constantly stressed. There is a way to take this guy and detoxify him. You use this enzyme called superoxide dismutase. This is the superoxide radical. And you can stick a couple of them together using this enzyme and go back to making hydrogen peroxide. Now, hydrogen peroxide is also somewhat toxic. It's not great to have around, but it's better than the superoxide. And because it's not great to have around, there are also enzymes that will take hydrogen peroxide and turn it back into water. So either catalase or peroxidase will do that. Having these enzymes means that even if you're using oxygen as your terminal electron acceptor and generating these dangerous chemicals, you have a way to deal with that. So if you're a microbe that is living in an oxygenated world, do you think that you're going to have one or more of these enzymes? Yes, no, maybe? The answer is yes. You're gonna, if you live in an oxygenated world, you're gonna have at least one of these. You're gonna definitely have this one. You might have these, just depending on what kind of chemistry you do. If you're living in an anaerobic environment, would you bother to make these? No. So depending on which enzymes you make, you can actually tell something about the organism's requirements for oxygen, even without having to do the test that I just showed you. This shows you sort of the breakdown of who has what, so you can read this on your own. All right, so I want to go into this next part, but I don't want to make this too long, and I also think you need a little break for me talking, so we're going to come back at 7:05 and resume.